All right, thanks for coming. I'd like to introduce Bryan Ayers. He's been working for the past 2 years on some, is it 3 years? Right, it's been so fast.
Yeah, whatever. Just trying to get me out of your hair.
That's right. He's been working on tick ecology and disease ecology. So he's going to tell us about what he found, so I'll let him take it away.
Okay. First off, thank you all for all coming here. I know a lot of y'all come from many places, or not so far. But here's our results for the research I've been working on for the last 3 years. The title of the research is the detection of spotted fever group Rickettsia and Borrelia burgdorferi in East Central Alabama via Amblyomma americanum, Rickettsia scapularis, and Dermacentor variabilis. So the last 3 scientific names labeled here are actually the ticks that are involved with our research here. So first up, let's talk about some ticks. So I'm glad ticks aren't this big. That would be awful. But anyway, ticks fall under the arthropod phylum. They actually have 3 different life stages. The first of which is larval tick. It's the larval tick. They will emerge. This is the tick that will emerge from the hatched egg, and they actually only have 6 legs. Now, once they molt into their second stage, the nymph, they will then grow their 4th and final pair of legs, bringing them to a grand total of 8 legs. And they retain those 8 legs to the final stage of adulthood. Now, as we all know and don't necessarily appreciate, ticks are bloodsuckers. They require blood in order to advance to their next life stage, in order to absorb nutrients. So we're going to dub them as hematophagocytes. The ticks that we're investigating in this research, as I said before, Rickettsia scapularis, which is known as the black-legged tick, or more popularly known as the deer tick, there's a picture of it in the top corner, and it's got more of a teardrop shape for its body. The Amblyomma americanum, our Lone Star tick, is located in the center there, and then Dermacentor variabilis, which is our American dog tick, and it also has a teardrop-shaped body shape. So, as far as where do ticks live? Well, first off, all of our research ticks are grouped into a category called the hard tick. And this only indicates that all these ticks have a very hard exoskeleton, increasing their survivability. But this hard exoskeleton, it requires to some degree, some moisture and humidity in the environment. So most of the species are going to prefer these environments. Pregnant females are going to require some percentage of leaf litter for laying their eggs. And then once those eggs hatch into the larval stage and then eventually into the nymph and adult stages, those nymph and adults are going to climb out of the leaf litter and onto lower to mid-growing vegetation to put themselves in a better position to attach to a new host. So as far as tick movement goes, they have 8 legs, they can move, they just can't move very far, very fast. So a lot of their distribution, their ranges for these different species, they require and rely on their host that they obtain their blood meals from. And their hosts can range anywhere from small rodents to mammals, small mammals, reptiles, and even larger mammals like you or me, or the white-tailed deer. Now, in previous research, it's thought that the white-tailed deer is one of the larger contributors to tick distributions. So, tick-borne pathogens and how they're involved in the life cycle of all of our ticks here for this research. Well, first off, where do these tick-borne pathogens originate from? All the pathogens that we're looking at in this research are caused by bacterial infections. And these bacteria will originate in what we call a reservoir host. This reservoir host, it allows this bacteria that resides within them to multiply and grow without restraint. And so what happens is a tick will come along, attach this reservoir host. As it ingests its blood meal, it also ingests the pathogen, and then the tick is considered what we call a tick vector. Now, a vector is any organism that can obtain, maintain, and transmit a pathogen to a new, previously uninfected host. So this mode of transmission is called horizontal transmission. I'll use this term a little later on, so put this in the back of your head for now. Now, the opposite of that, on the flip side of that, we have vertical transmission, which in our case, a female tick would ingest a particular pathogen and then transmit it down to her offspring as she is fertilized. So the bacteria that are involved in our research, we're going to be looking at Lyme disease. Of course, everyone has probably at least heard of this disease, and then a group of bacteria called the spotted fever group Rickettsia. We'll get a little more into these in a second. Lyme disease is the causative agent, or has the bacterium that is the causative agent for Lyme disease is Borrelia burgdorferi. And it has been dubbed the most reported tick-borne disease in the United States. Now here's a map the CDC has produced. The darker areas, the darker blue areas, are areas that are more concentrated with more cases for Lyme disease. So you can see that the states, the northeastern states and the northern midwestern states typically have a higher density of cases than the rest of the country. We have more cases on the west coast and the southern United States, but not nearly as many as those previous regions. So, Burgdorferi, what does it look like? This particular bacterium is classified as a spirochete bacterium. It is gram-negative. It has a periplasmic flagella, meaning that it utilizes this flagella for added mobility. And then it also has these outer surface protein receptors that line the entire outer membrane of the cell. Now these receptors are very important. These receptors are utilized by the bacterium for kind of like a line of communication. It identifies, reaches out and grabs a hold of neighboring cells, identifying what cells, right, if those cells are food, if they're friend, if they're foe, whether they're a potential host cell, all that kind of stuff. So a line of communication. Now, how is it transmitted, this Burgdorferi? It is, like I said before, it's transmitted horizontally. And so, therefore, it originates in a reservoir host. More than likely, it's going to be the primary host, or the primary reservoir host in these areas is the white-footed mouse. By no means is this the only reservoir host, but it is one of the more largely used hosts. So, tick grabs onto a reservoir host, ingests the pathogen, and then is able to transmit it to its next available victim. So, now as far as tick vectors go, Ixodia scapularis are black-legged tick, or as some of y'all may know it as the deer tick. This is considered the primary vector, or one of the primary vectors, I'm sorry. Let me retract that. It's one of the primary vectors. By no means is it the only vector for this pathogen. So what does it do once it's inside of you or me, once we're infected? Burgdorferi will begin to multiply at the invasion site where the tick bit you, and then it will expand to any kind of blood vessels and the bloodstream to kind of utilize your circulatory system as a highway to infect potentially useful host tissue. Now this bacterium is going to prefer to be in host bacterium, or is going to prefer to be in host tissue as opposed to the bloodstream. So it does not stay in the bloodstream very long. And it's going to prefer host tissue such as nervous system tissue, heart, and joint tissue. Okay, so our spotted fever group, or SFG, this is one of 3 subgroups within the Rickettsia genus. And of this group of bacteria, there are 9 members that have been found in the United States. Now, of these 9, 4 have been identified as pathogenic, and they include Rickettsia amblyomyi, Rickettsia parkeri, Rickettsia acari, and Rickettsia rickettsii. Now, the last one, Rickettsia rickettsii, is better known as Rocky Mountain spotted fever. Now, when Rocky Mountain spotted fever is compared to the other 3, as far as virulence goes, Rocky Mountain spotted fever is hands down one of the more virulent tick-borne pathogen bacterial infections when compared to the others. So, in our ticks, for our research, at least one of these pathogens has been identified as naturally occurring in all of our ticks. So, what does the average member of this group, spotted fever group, look like? They're typically going to look bacillus-shaped, meaning they're going to kind of look like a pill or like a hot dog we see there in the picture. And they're going to be gram-negative. They're going to be non-mobile, meaning they don't have any flagella or cilia to utilize for movement. So wherever the water's going, they're going that way too. And they utilize our outer surface protein receptors again to reach out and identify if neighboring cells are friend or foe, but more importantly, whether a cell is a potential host. So, how is it transmitted? It's transmitted horizontally, like we discussed before. The tick will bite you, or once the tick ingests the pathogen from a reservoir host, it will then transmit it to a previously uninfected host here. Now, once it infects you or me, what happens then? Since this particular bacteria, or this group of bacteria are non-mobile, from this point on, we're just going to talk about the pathogenic members of this group, as opposed to the non-pathogenic. But these pathogenic members of the spotted fever group are non-mobile, so therefore they're going to highly utilize the bloodstream for movement or identify potential host cells. Now, the preferred potential host cell is going to be what we call the endothelial cells. Now, these endothelial cells line any of your cardiovascular system. They are the inner lining of any of your vessels within your circulatory system. And they're going to prefer to host these endothelial cells located more around the heart, the nervous system, brain, and lungs. So once one of these pathogens in this group locate and identify a potential host cell, it will force the host cell to engulf the pathogen, and then the pathogen will begin to absorb any kind of nutrients to utilize for growth and multiplying, eventually killing the host cell and allowing the pathogen, the new now multiplied pathogens, to spread to other host cells. So, our objectives for this particular research: first off, we went out and collected ticks at previously identified sites. We extracted DNA from each of our tick samples, ticks that we collected, and then we utilized a highly specific, very unique protocol called real-time PCR, and this was used to identify if there were any of these pathogens present or absent within our samples. Also, lastly, we identified any kind of ecological factors that might be contributing to the absence or presence of these pathogens. So first up, materials and methods. How do we go about conducting this experiment? Well, first off, we identified 12 different sites that were located within Fort McClellan and also within the Mountain Longleaf National Wildlife Refuge just down the road next to Anniston. And of these 12 sites, we divided them into two different categories. The first category, which 9 of our sites fell into the urban category, and this urban category was defined as any site that was located within 300 meters of the nearest clearing. Now, our second category, which our remaining 3 sites fell into, was our forested category, and it was defined as any site that was at least 1 kilometer away from the nearest clearing. So, once we identified our sites, we then marked out a 20 by 20 meter plot to obtain all of our data for vegetation and tick collections. So here is an aerial picture of all of our sites. Each yellow dot represents all of our sites that we collected from. And once we identified all of our sites, the first thing that we did was we identified any kind of vegetation we took some vegetation data. Actually, 2 different types. The first one is called basal area, and it determined the amount of area of trees at each site. The second vegetation data that we took, type of vegetation data we took, were cover calls, and this pretty much looks at 7 different vegetation types. Those vegetation types are sapling, seedling, shrub, fern, herb, vine, and grasses. And for this data, we identified the percentage of ground cover that each one of these vegetation types was found at each site. Between 2 different sites. We also use multi-response permutation procedures. We'll shorten that down to MRPP. And it identified not only significant differences between 2 different variables, but also heterogeneity between those variables. Third, we used our Pearson's correlation, which identifies any kind of relationships. It identifies negative or positive relationships between an independent and a dependent variable. And then lastly, we use our regression analysis, which identifies the strength of any linear relationship between 2 variables, an independent variable and a dependent variable. So, a lot of people ask, how do you collect a tick, Bryan? So, for this technique, we utilize a technique that utilizes the methods that ticks use to obtain a host. And the way that they do that is they are attracted to any kind of organism that is exhaling CO2. So just by you or me going out in the woods and breathing like normal as we're walking through, they're going to be at least alerted to our presence. And so they're going to put themselves in a position to hopefully get attached, put themselves in a position where they can attach to you. So we utilize this behavior, and I, as many of y'all have seen, I put on some white overalls that I could see all of the ticks that might get on me as I'm walking through the woods. But at each one of our sites, we use two different flagging methods. The first is called, like I said, the flagging method, and it's basically two different flags that we used. The first one, like I said, is the flagging method, and there's a picture of this type of flag on the top. This is real sophisticated science here. We got a flannel cloth that is attached to a wooden rod, and this method of obtaining ticks, it obtains typically our nymph and adult ticks, and we drape this cloth over any kind of small to medium-sized vegetation where these nymph and adult ticks will reside typically. Now, our second method that we used was the dragging method, and there's a picture of the flag that we used down there on the bottom, and it's basically a wooden rod with ropes coming off of it that then attached to a flannel cloth, and this method allows the entire flannel cloth to drag along the forest floor where all the leaf litter is, and potentially where all of our larval ticks are going to reside. So, with both of these methods, we're going to check each side of our cloth every 10 to 15 paces as we kind of randomly walk through our site. And we're going to do that for 10 minutes with each flag. So we'll stay at each site for a grand total of 20 minutes. And we collect it at each site at one time a month for 8 months, between the months of from the months of March through October and in the year of 2011. So here's a picture of me using the drag method here. Now, once we obtained our ticks and submerged them in isopropanol for preservation, and then we brought them back to the lab, we then wanted to extract any and all DNA that resides within the tick. Because hopefully our DNA extraction process does not discriminate. It will actually obtain any DNA, whether it be the tick or hopefully some DNA that is associated with with our tick specimen. So this process, it will obtain any and all DNA, like I said, whether it be tick or bacterial. And so what we do is we use this extraction on each individual nymph and adult tick, and we'll grind each one of these ticks up using a sterile homogenizer. Now the larval ticks, they are about the size of a pinhead. They're really tiny. And so instead of grinding up each larval tick individually, we put them into groups of 10, this is what we're going to call a pool. So one pool equals 10 larval ticks. And we ground each pool using, again, a sterile homogenizer. After all this grinding and homogenizing, we then used a commercial extraction kit from the QIAGEN company. So after we extracted all of our DNA, we then wanted to see if our DNA preps actually went well and we actually collected DNA. So our thought process around this was, if we're extracting DNA from these ticks and bacteria, obviously we know the ticks are in there. We don't necessarily know if any of our bacteria are in there. So what we're going to look for, since we know the tick's DNA is in there, we're going to look for our tick DNA. So we utilized a procedure called conventional PCR, and it identified a particular sequence within the tick genome. And if our sample actually had the tick DNA in there, then our sample was considered positive, and we could use that extraction for further research. So we utilized a positive and negative control along with this. Now, the next step that we used was, like I said before, is a very highly specific and unique procedure that can identify individual species of bacteria. And this process is called real-time PCR. It utilizes a molecule that we have named the dual-label probe. And this probe, it fluoresces whenever it's in the presence of the specific bacterial DNA that we're looking for. So in our case, if we have a sample that has the DNA from one of our pathogens, and we're using our dual label probe, that dual label probe will then emit at least some form of fluorescence, indicating that that sample is indeed positive. So we utilized a positive and negative control along with each of our samples here. So for our first real-time PCR protocol, that we utilize this PCR protocol on Borrelia burgdorferi. And this procedure we actually developed here at JSU on campus. We put all the man hours into it, beat our heads against the wall until we got it right. And we came up with this procedure utilizing a forward and reverse primer, along with, again, our dual label probe that fluoresces whenever the DNA from this particular pathogen is present within our sample. And this pathogen, or this procedure, actually identifies a 79 base pair sequence within the DNA of this bacterium. So our second real-time PCR protocol that we utilized was for our spotted fever group, of course. Now, before we executed this protocol, we, instead of utilizing this protocol on each individual tick, we actually ended up using this protocol on our pools of ticks. So what we did was we created pools of ticks for all of our DNA extractions, adult, nymph, and larval ticks. And in each pool, we actually had 10 DNA extractions within each pool. And then we utilized this real-time PCR protocol, like I said before, on these pools. So this protocol, again, utilizes the dual label probe that fluoresces whenever the bacteria is present from this group. And it actually identifies a 74 base pair sequence within the genome. Now, this process, when compared to the Burgdorferi protocol, the Burgdorferi protocol will actually identify the individual species, the actual Borrelia burgdorferi species, whereas this particular protocol only identifies members of this spotted fever group. It does not identify species-specific individuals. It only identifies whether they're a group or a member of this spotted fever group. DNA extractions within each pool. And then we utilize this real-time PCR protocol, like I said before, on these pools. So this protocol, again, utilizes the dual label probe that fluoresces whenever the bacteria is present from this group. And it actually identifies a 74 base pair sequence within the genome. Now, this process, when compared to the Burgdorferi protocol will actually identify the individual species, the actual Borrelia burgdorferi species, whereas this particular protocol only identifies members of this spotted fever group. It does not identify species-specific individuals. It only identifies whether they're a group or a member of this spotted fever group. So, results and discussion. What happened and why did it happen that way? Well, first up, our ticks that we collected. We ended up collecting about 1,040 ticks, so a lot, but it would have been nice to have more, have a little bit more data, a little bit more, anyway. We ended up collecting 3 different tick species, as we talked about before, our Lone Star tick, our black-legged tick, and our American dog tick. Now, as you can see, according to our pie chart and our numbers here, the Amblyomma americanum, our Lone Star tick, it was the vast majority of our collection. Actually, it was about 1,021 individuals that we collected, followed by the American dog tick, which only had 14, and then the black-legged tick with only 4. So, like I said, 94% of our collection consisted of Lone Star ticks. The blue right here in our paragraph indicates all the Lone Star ticks. So, large portion of our collection. Now, these are the numbers of ticks that we collected each month during the 8-month period. And as you can see, the blue represents all the Lone Star ticks, and the red represents all the black or the American dog ticks, and then the green here, only in March, that's when we had our Lonely 4. And as you can see, in July, August, and September, this is when the majority of our ticks were collected. So, what can we deduce and conclude about these ticks that we collected? Obviously, the majority of our collection were Lone Star ticks, Amblyomma americanum. So we can loosely imply that most of the population in this region, Fort McClellan and the the refuge that we collected from, probably most of the ticks that you're going to collect are going to be Lone Star ticks. Now, as far as some reasons why maybe this data might have been skewed a little bit, is the fact that previous research has found that the Lone Star tick, when compared to Ixodia scapularis and Dermacentor variabilis, the black-legged tick and the American dog tick, when the Lone Star is collected to these ticks, then the Lone Star tick is actually then considered more aggressive in hunting and identifying and hunting out its potential host to obtain a blood meal. So it'll run a little faster, it'll reach its arms out a little further just to grab that potential host. Whereas these may not move quite so fast, whereas with vigor. So our statistics, stay with me here. The first up, we use our Mann-Whitney U-test. And like I said before, this identifies any kind of significant differences. And in our case, we looked at the number of ticks collected as our first variable, and then the distance of each site to the nearest edge, or the nearest clearing for our second variable. So, like I said, this identifies significant differences, and what we looked at here are the sites that are further away from the nearest clearing. We're looking to see if there's any significant differences between those sites and those sites that are closer to the nearest clearing. And our p-value that is generated as a result of the statistic, if our p-value is less than 0.05, then we have a significant difference, and ours definitely is at 0.0002. So we can say that those sites that are further away are significantly different from those that are closer. Now, MRPP, this is a highly specified statistic that not only, again, identifies significant differences between variables, but also heterogeneity. Heterogeneity meaning that there is a difference between two different variables. So in our situation, we looked at the same variables as Mann-Whitney, and we looked at the number of ticks collected versus the distance of each site to the nearest edge. And for this particular statistic, we identified another p-value again, and like I said, if the p-value is less than 0.05, then our variables will be significantly different. So our p-value is significantly different at 0.011, and this identifies that, yet again, our sites that are further away from the nearest clearing are indeed significantly different from those sites that are closer to the nearest clearing. Now, not only did it look at that, but it also looked at those sites within each category, our forested category and our urban sites that are within that category. And it identified that the forested sites and the urban sites, when comparing those sites within each category, there is some sense of homogeneity between those sites. And also, it looked at those categories and compared those categories to one another and determined there is at least some degree of heterogeneity between those different site types. Okay, so our third statistic, Pearson's correlation. Like I said before, it identifies relationships, positive or negative, between an independent and a dependent variable. And we looked at three different situations. The first of which is, well, for each situation, the dependent variable was our number of ticks collected, and then our independent variable was different for each. It was number of ticks collected versus the basal area, the the distance to edge, and the percentage of each cover call to the number of ticks collected. And the only data that we really found of any interest using this statistic was found within our vegetative cover calls. Now, some highlights here, the first of which is the percentage of shrubs. We found a positive relationship between the percentage of shrubs found at all of our sites during the month of July and the number of ticks collected. Another one for the percentage of ferns at all of our sites during the month of August. Our percentage of herbs at all of our sites during the month of April, and then a negative relationship between the percentage of grasses at all of our sites in the month of June. Now, our fourth and final statistic, almost done, just stay with me. Regression is the regression analysis. And the regression analysis, like I said before, identifies the strength of a linear relationship between an independent and a dependent variable. We looked at the same three situations as with Pearson's correlation. Yet again, the dependent variable being the number of ticks collected in all situations versus the basal area, whether these sites have any kind of heterogeneity. And we identified that not only are the urban sites more fragmented, meaning that those forested sites within the sites within closer to urban areas rather, they're going to have roads and fields and maybe some housing developments, causing these areas, these urban sites, to be more fragmented, more patchy. And not only did we determine that, but also the forested sites, they are more secluded, meaning they don't have any roads near them, they don't have any houses, more or less they don't have a whole lot of people going by these sites. And so from previous research, we've identified that in these urban sites that are fragmented, these fragmented sites are going to support more potential hosts and reservoir hosts for ticks to attach to at these urban sites. Now, our only negative relationship that we've identified here are that the tick densities are typically lower at sites that have a higher grass percentage, especially during early summer, like in the month of June. And the reason for this is that grass, it is semi-nutrient, or has some nutrients to it that a lot of organisms can consume, but there are better things out there. And at this time during the year, there's going to be a lot of other herbaceous food items that are going to be more preferred and more desirable. And so some of these potential reservoir hosts that are grazers, they're going to flock to these sites that have the more preferable food items, whereas these sites that have just grasses, not going to have too many visits from these hosts. So we have less hosts and reservoir hosts visiting these areas, less ticks are going to be identified here because they are contributing to a lot of tick densities and ranges and distribution. One example could be our white-tailed deer. So for our first positive relationship here, sites that were typically higher in percentage of shrubs typically had a higher density of ticks. And due to this, the shrubs, a lot of times organisms, especially our host and reservoir host potential organisms, are going to flock to, some of them are going to flock to these shrubby, higher percentage shrub sites due to the fact that these shrubs are giving them natural cover within their environment to not only evade from predators, but just give them a place to maybe take a nap during the day. And so if these potential hosts are visiting these sites that have a higher percentage of shrubs, ticks are going to have a higher ability to attach or let go of these hosts, giving a higher density for these ticks at these sites. Now, our last positive relationship that we have identified is that typically with higher tick densities, or we're going to find higher tick densities at sites that have a higher percentage of ferns. Now, first off, ferns, as many of y'all can probably imagine, ferns, typically most species are going to reside at more moist, humid environments, and especially in August when we found our findings here, interesting findings. These ferns, if they're growing well at these moist, humid environments during August, it's really hot. There's not many of these sites left around because everything's so hot. And so if these ferns are growing in these moist, humid environments, a lot of other herbaceous foods that are maybe more preferred by the potential host, they're going to be growing at these sites as well. And so these potential hosts are going to be flocking to these sites that have more of these ferns and herbaceous foods available for them to eat. So this is going to be a reason why, or give ticks ample time to attach or release from these potential hosts. For example, again, our white-tailed deer. Now, moving on from statistics, you can all breathe now. The DNA confirmation that we want to identify how many of our DNA extractions actually went well, that we obtained DNA from. And from our 314 DNA extractions that we conducted, 8 of those samples ended up being negative, so we did not extract any kind of DNA. Sad day, but in retrospect, looking at the sheer number of extractions that we had, it's not a huge deficit there. I would like to have those back though, that would be nice. So, but anyway, we ran a positive and negative control along with those to identify whether the samples were positive or negative. Now, the results for our real-time PCR protocol for Borrelia burgdorferi, none of our samples actually ended up being positive, so yay, no Lyme disease. So none of our samples ended up being positive. Along with that, we utilized a positive and negative control to identify our results for each of our samples. So making some sense out of this too, since we did have no positives, some reasons why this could be the case, reason why this might have happened, is like we said before, one of the primary vectors for this particular pathogen is Rickettsia scapularis, our black-legged tick. Now, if you think back to the number of ticks that we had, or number of black-legged ticks that we had, it was a very, very small number. It was only 4 out of a lot. And this particular tick in previous research has been identified as being really efficient at maintaining and transmitting this pathogen. So since we had a lower number in our collection of this particular tick, it's not the dominant tick, then this pathogen doesn't have a whole lot of options as far as being transmitted. Now, on top of that, like I said, the black-legged tick is not the only vector, but is more efficient in comparison to Amblyomma americanum, which is our Lone Star tick, because it can harbor this particular pathogen, but not for very long. It can only harbor it for only a few days before the bacterium actually dies within the tick. So it's not a whole lot of time to transmit. Now, on top of that, Burgdorferi, as we talked about before, it is horizontally transmitted, and it is since it can't be vertically transmitted, then this might reduce some of its fitness within the ecosystem because it only has one mode of transmission. Now, our results for protocol, we ended up creating 32 pools, and of those pools, 30 ended up being positive, whereas 2 were negative. So pretty good. Vast majority of our pools tested positive, and along with that, we utilized a positive and negative control to identify which samples were positive and negative. Now, along with that, with our results, we identified what we call a minimum infection rate, and this infection rate is we identified a 9.4% of all of our ticks possess, at least this is a very conservative number, 9.4% of our ticks possess a member from this group. Now, remember that, like I said before, this particular protocol for identifying members of the spotted fever group only does that. It only identifies members of this group. It does not identify pathogenic members or non-pathogenic members, much less on the species level, individual species. So due to that, most of these members have been considered endosymbionts, meaning that they require some host in order to survive during some point of their life cycle. And so a lot of these positives may be a result of some of those endosymbionts. And on top of that, most of the members of this group, like I said, can be transmitted not only horizontally but also vertically, so from the mother to her offspring. And we do have an example of that within our research. The pools of the 30 pools that were positive, 10 of our pools solely consisted of Lone Star larvae, meaning that larvae at this stage, when they're first hatched, obviously they haven't had a blood meal yet. And so they haven't had the opportunity to be exposed and become a vector for this pathogen, for one of these members of this group, but yet they have it. So we are identifying some vertical transmission that's going on from the mother to the offspring. So in conclusion, we can all breathe? Yes. In conclusion, the white-tailed deer here is going to be assumed, or it has been assumed, like I said, in previous research to be one of the major contributors to amblyomma, to not only tick distributions in general, but also the Lone Star tick, Amblyomma americanum, and their mobility and range, and also, more importantly, within these urban patchy sites that we have identified as our urban category. Now, our second point is that Borrelia burgdorferi, even though we did not have any presence within our samples, this does not necessarily rule out the fact that we might have some of this pathogen located and residing somewhere around here in this region. It's just not very probable to find it. Also, we had our vast majority of our specimens come up positive for members of the spotted fever group. Now, like I said before, this does not indicate that all of our positives indicate pathogenic members of this group. It just identifies members that are part of that group. So, that being said, some future research that we could maybe pursue, I probably won't because I won't be here, but some future research that we could look at from an ecological standpoint, we could collect and compare leaf litter weight at all of our sites and the number of ticks that are collected at those sites, the tick populations. Basically, like I said before, ticks female ticks lay their eggs in the leaf litter, and the larvae tend to stay in that litter for the remainder until they actually molt into a nymph. So this could be highly beneficial. Another thing that we could do is collect at all of our sites for the full year, because some ticks may continue to be active past our parameters of the dates that we collected from. And also we can identify and utilize a real-time PCR protocol that will identify not only pathogen versus non-pathogen within this group, but also species specific. So, thank you all for being here. I want to especially thank Dr. Robert Carter, Dr. Chris Murdoch, and Dr. Benji Blair for being my advisors and guiding me towards graduation. And then also I want to thank all my colleagues here at JSU, Daniel Wright, Andrew Dotson, Richard Watkins, Bryan Ingram, and Matt McCurdy for helping me through this process. And then also the JSU Biology Department for allowing me to do this research, and then Jacksonville State University for allowing me to be a student. And then lastly, thank you all, my friends and family, for putting up with my solitary lifestyle that I've recently acquired. So, yes. So, thank you all. Okay, so any questions? Yes.
I have 8 million questions.
All right.
I'm going to limit it to 2. So, on your positive control, are those just obtained from standard? Do they ship those to you? Is there some sort of bacteria you can just?
Actually, yeah. For our Borrelia burgdorferi positive, we actually obtained that from a guy named Kelly Clark at the University of North Florida, right? Okay. And that was in culture that we were able to pull that.
Those cultures are southern. Those come from ticks that are collected. So there's southern.
Okay, so those ticks were collected in the south, so obviously that strain has been identified here in the south. And then the spotted fever group, that was obtained from Dr. Goddard at Mississippi State, who he actually sent us bacterial cells of the actual spirochete, and we isolated the DNA from there. So yeah.
And then the second question is on your analysis to determine if the sites are actually between urban and forested, if the within and the between variability
Right.
is heterogeneous or homogeneous. Did you use just your plant cover? Was that the variables that you put in to figure out?
We actually, we just looked at the distance from each site to the nearest edge, and then number of ticks collected.
Okay.
So those were the only variables that we looked at there. So.
Cool. I'm done.
Yes, sir.
Is real-time PCR more specific than a standard PCR protocol, and if so, why?
How? Why? Okay. Yeah, the standard protocol that he's talking about is, I briefly mentioned it, it's called conventional PCR. Real-time PCR is very specific. It utilizes, like I said, this dual-label probe that actually fluoresces. In the conventional PCR protocol, it does not use this probe. It does not emit any kind of fluorescence. You actually have to utilize what we call gel electrophoresis to actually identify positives. So not only is it a time saver for real-time PCR, because you can identify positive or negatives right then and there in one reaction, but it is highly specific because not only do you have a reverse and forward primer that are amplifying the target DNA, but also the process won't work if the dual label probe doesn't sit down in the right spot in the middle of the target sequence. So if the target sequence isn't there, then no fluorescence occurs.